Well, welcome back. Now we're going to take a look at the evidence that the NMDA receptor is involved in the generation of LTP. So there is our topic. Now, if you recall from last time, this is the setup, the last lecture. This is the setup that we used to demonstrate LTP. What we want to now look at is what's going on during the tetanus in this synapse that's leading to LTP. It's making the synapse stronger so that it leads to a stronger postsynaptic response. Now, to do that, I'm going to show you a bit of more detail here of this membrane, the postsynaptic membrane. There it is. And what I'm showing now are receptors. These are so-called AMPA receptors. They are receptors for the neurotransmitter glutamate. And when glutamate is the neurotransmitter released by the presynaptic component of this synapse. When glutamate is released, it binds to the receptor, activating it, the AMPA receptor. And the AMPA receptor is in fact an ion channel. When it's activated, it opens and sodium flows into the postsynaptic side. Thus we have a depolarization. Okay, so if we just had one of these simple stimulations here, a little bit of glutamate is released, a little bit of sodium comes in, and this depolarization, the sodium ions, they diffuse to the cell body, and the depolarization is picked up here by the recording electrode as an excitatory postsynaptic potential. I'm going to show that here now. Here is before the neurotransmitter is released. With the release of the neurotransmitter, depolarization here, and we're now measuring an excitatory postsynaptic potential. So the EPSP. Good. Now let's look at what happens during one of these, during the tetanus. During the tetanus, you give a high frequency stimulation here, so you get these high frequency action potentials charging along the Schaefer collateral here, and what happens here in the synapse, you get a very strong release of glutamate. So far higher concentrations are released, and consequently many more of these AMPA receptors on the postsynaptic side here become activated. A larger amount of sodium flows in, so the depolarization is stronger, and what we're measuring here, we can see it, the depolarization, stronger than EPSP, and in this case it's actually reached what's called threshold, and at threshold the depolarization here activates so-called voltage operated channels here in the cell body, and this generates an action potential. And this action potential, of course, it goes along the axon and ultimately will lead to neurotransmitter release somewhere else in the brain. Now, a special property of neurons that are involved in learning and memory is that they have the capacity to carry this strong action potential not only along the axons but also back into the dendrites. And for this reason, because it's going back into the dendrites, it's called back propagating action potential. And it just goes up all the dendrites, causing a strong depolarization. So here's our membrane here in the synapse, and because of this back propagation, we have a very strong depolarization. And now, this is during the tetanus. Something very, very special is now going to happen. I mentioned that we have all these AMPA receptors. They're in all these different synapses here. As well, all these synapses possess another receptor for glutamate, and this is called the NMDA receptor. So we have NMDA receptors here as well. Now, during the tetanus, these NMDA receptors become activated. But they become activated only during the tetanus. The reason for this is that these receptors are so-called coincidence detectors. Two different events must occur in order for the NMDA receptor to activate. The first event is the presence of neurotransmitter. Well, it certainly is present here during a tetanus. In fact, there's high amounts. The second event is that there must be a strong membrane depolarization. Well, that is taken care of through this back-propagating action potential. With these two criteria met, the NMDA receptor opens and we have calcium flowing into the cell, because NMDA receptors, they are highly specific for the ion calcium. So, coincidence detection. And then the calcium, that activates events, and I'll go in a little detail a little later, but this activates events inside the neuron, inside this neuron here in the postsynaptic side, activates events that ultimately leads to this long-term potentiation, which is basically a strengthening of this synapse. Now, in this case, it's specific for this synapse. Here's another synapse up here being innervated by another Schaefer collateral. In this neuron, you're going to find no LTP if you could measure it. Reason here is that the two criteria haven't been met. It is being exposed to this back-propagating action potential, so it has strong membrane depolarization, but there is no glutamate here in the synapse. Consequently, there will be no activation of that NMDA receptor. So only this synapse will become strengthened, undergo LTP. Okay, now what I want to do is take a look here in more detail at the NMDA receptor to see how can it act here as a coincidence detector. Now, like most ion channels, the NMDA receptor is constructed from various subunits. These are separate proteins. And in fact, for the NMDA receptor, it has 5 subunits. 2 of these subunits actually contain a binding site for the neurotransmitter glutamate. I'm going to take this subunit away now, and you're going to see something very special about the NMDA receptor. Namely, there is the ion magnesium situated right here in the ion channel where the calcium should be flowing. And what this magnesium actually does is blocks the channel. So here is our glutamate. Glutamate can bind to the receptor. Now if this was an AMPA receptor, this would be enough to activate the receptor. So, and with the AMPA, of course, that's a sodium channel, so you would have sodium running into the cell. But this is an NMDA receptor. Glutamate is bound, but nothing happens. And nothing happens because it's being blocked. To lift that block, you need this strong membrane depolarization. And what happens then with this depolarization is that the magnesium is kicked out or leaves the channel, and thus the channel is now open and available for the calcium flow. Now, what is the evidence that the NMDA receptor is involved? Well, I'm going to discuss here 2 pieces of evidence. The first piece of evidence I'm going to describe is experiments with so-called receptor blockers. Receptor blockers, those are chemicals. AVP is one. This is an abbreviation for a particular chemical. Name is not actually important. What is important to know is that AVP is a NMDA receptor blocker. What happens, it binds to the glutamate. In fact, its structure is rather similar to glutamate. Only APV is incapable of activating the NMDA receptor. So even if you have a depolarization, magnesium leaving, still it can't activate. Only glutamate can activate it. Thus this is called a receptor blocker. So no receptor activation in the presence of APV, if it's of course in high enough concentration that it's competing highly successfully with the glutamate for the binding sites. Now here is what you saw in the last video, our demonstration of LTP with a stronger strength of the synapse of the excitatory postsynaptic potentials following the tetanus. This same experiment can be done, only a so-called puffer is put into the equipment so that you can actually squirt on some AVP during the experiment. And here is where it is put on just before and during and just after the tetanus. So at this time period we're blocking the NMDA receptor, and what we will see before the tetanus, and now here is after the tetanus. And as you can see here, the LTP is blocked. Now, can we block learning with AVP? So this is in vitro. If we go into the living animal, in vivo, into the intact animal, and inject AVP into the hippocampus, can we block learning? Well, those experiments have been done as well. So each time before the trials, a little bit of AVP is brought with a cannula into the hippocampus. And what you can see here, this is the latency period, how long it's taking the animals to find that hidden platform in the Morris water maze. And you can see that the control animal is outperforming the AVP treated animal in every case. So blocking the NMDA receptor, it blocks LTP, and what you see up here, it also has a dramatic effect on inhibiting learning. Now, here is what some people consider the ultimate proof, a so-called gene knockout. So here we have our genetic engineers, and if you've ever been walking around in a neuroscience department or a biology department and you see a lab with lots and lots of test tubes and little bottles, that's almost certainly a molecular biology lab. And in these test tubes, oftentimes what you have is gene constructs that have been manipulated by the various chemicals that you're finding in there, so that you can mutate these genes. Now, as you know, genes, that's found in your DNA, and the DNA is then transcribed to messenger RNA, and then you get translation to protein. And what these genetic engineers can do is they can make non-functional genes by having the genes in vitro and then treating it in certain ways, usually with the various enzymes, and introduce sort of mutations in these genes so that they're coding for a non-functional protein. And then these genes can be in various ways that I don't have time to go into. They can be introduced into embryos, and ultimately you can produce a transgenic animal which has this altered gene, this non-functional gene. Now, for the NMDA receptor, one of the subunits, one of the critical subunits for the NMDA receptor was mutated, and a transgenic animal generated. So this is our so-called NMDA knockout mouse. And the mouse, in fact, looked perfectly normal. It acted perfectly normal. But, and here's the big but, let's take a look first of all here at the behavior on the LTP. Here's the control in blue, and here is our knockout mouse. This is a symbol for the knockout mouse. What this is indicating is, in fact, the NMDA receptor has been specifically knocked out in a particular area, so-called CA1 region in the hippocampus. And you can see this has had a dramatic effect on the LTP. Almost no LTP is generated at all by knocking out the NMDA receptor. And if we look here, this is again, this is on the in the intact animal on the Morris water maze, and you can see here performance of the CA1 knockout far worse than the control animal. So again then, lower LTP, and you see in the intact animal a lower performance in a memory situation. So we can really put a dunce cap on this particular transgenic animal. Question: if lack of an NMDA receptor makes you stupid, will NMDA receptor overexpression make you smart? Well, the molecular biologists have looked into that as well. They have produced a mouse which they called Doogie. And what this is, they've introduced extra NMDA genes, and so you get actually an overexpression. And again, the way they did it, they manipulated so this NMDA overexpression came primarily in the hippocampus. Now, they named the animal Doogie. And Doogie apparently comes from an old American television series called Doogie Howser, MD. Doogie Howser, he was a precocious teenager who had already at age, I don't know, 15 or 16, had obtained his medical degree. So he was a medical doctor. Now, the mouse Doogie, not quite that smart. Nonetheless, he shows stronger LTP. This is just another way of showing LTP. Here is in this case, the Doogie is in green. I think I've indicated it, yeah. So here's a stronger LTP compared to our so-called wild type mouse. And here it is here with the Morris water maze, finding that hidden platform. Doogie at almost every point here is significantly higher in significantly less time in finding that hidden platform. So Doogie is smarter. And in fact, the week that this particular article came out, this was the cover that week in Time magazine with the question here, have the neurobiologist found the IQ gene? Okay, so a little summary now. Here we have the glutamate. It binds to the receptor, but nothing happens. However, if we have a coincidence of this binding with, at about the same time, a very strong depolarization, the magnesium leaves, and we have calcium flowing in. From there, the calcium goes on to activate enzymes inside the neuron. And these activated enzymes go on ultimately to induce long-term potentiation. Now these enzymes, most of them are in fact a class of enzymes called kinases. And kinases control the activity of other proteins. And in controlling the activity of other proteins, they can ultimately lead to LTP. And now some of these processes are very fast because you've already seen that this LTP, it occurs within seconds of being hit with that tetanus. Other changes occurring in these neurons involve the expression of genes, the new expression, new proteins being produced. So this all goes to contribute to LTP. Now this is called a signal cascade. I'm not going to go into any details here on all the various steps, but this is just the general idea. Because it's downstream from the so-called NMDA receptor, it's often referred to as a downstream signaling cascade. And others refer to it as an intracellular signaling cascade. Now this LTP, you've seen LTP, and this is a general statement now for any manipulation you give the animal that increases LTP, such as in our Doogie. This is always, I can't think of any exception, this is always results in an animal that has a better memory. Likewise, any treatment you give an animal that decreases LTP, we've seen two examples here, the receptor blocker and also the gene knockout. This inevitably leads to worse memory. And because of this observation, LTP is generally considered to be driving the memory. And in fact, other experiments, whole animal experiments, have actually shown that there is LTP in the hippocampus of the intact animal when it is learning something. Now, I don't have time to go into those experiments, but this really is dogma. LTP is driving force for memory. Okay, so that is then the evidence, a little bit about what's going on in that postsynaptic element, and the evidence that it's the NMDA receptor that is involved here in initiating the generation of this LTP. Now, in the final video, I'm going to discuss with you a bit about how you can make a memory with LTP. Okay.